What if the answers to our biggest questions, such as healing diseases, surviving in space, or creating future technologies, have already been solved by nature? From the stickiness of a gecko's foot to the antifreeze in ice fish's blood, in giraffes' massive hearts to ensure blood is pumped throughout their bodies, nature has been writing the code for survival. And now, scientists are learning to read it. It's all written in DNA, nature's ultimate blueprint. And through genomics, we're unlocking its secrets. Today, we're going on a mission to uncover the power of genomics. You'll meet scientists working on cutting-edge research, discover how genes shape life as we know it, and see how nature's designs are inspiring the future of medicine, tech, and even space. Welcome to Nature's Blueprint, virtual field trip. Hi, I'm Sloan, and I'm a student from right here in San Diego, and I can't wait for what we'll uncover today. But before we dive in, we need to lay down a few basics. Like, what is DNA exactly? What makes you different from me? And how are scientists using genomics? To help us break it all down, we're going to meet Chris, a senior scientist at Illumina who knows all this stuff inside and out. He'll talk us through the building blocks of life and explain how we can learn about genomics, the study of DNA, from ourselves to plants and animals, and even to outer space and beyond. Come on, let's go check it out.
Hey, Sloan.
Hi.
Welcome to Illumina.
Thanks for having me.
Thanks so much for coming. Here we develop DNA sequencing and technology that allows us to deepen the impact in the field of genomics.
And what is genomics in DNA exactly?
Genomics is the study of heredity, or how genes are passed down from generation to generation. And DNA is the molecule that makes up genes.
And what is DNA made up of?
DNA is the molecule that encodes all of our genetic information. You can kind of think of it like the alphabet. So depending on the sequence of those letters, they can comprise a different word. And that word has different meanings depending on that sequence of letters, right? So your height, your facial features, really everything about you that you can see and a lot of things you can't are encoded in genetic information. Humans are 99.9% similar to each other genetically. So everything you can see in people and all the diversity in the world is explained by 0.1% of genomic differences.
So my physical features, like my eye color, are passed on from my parents, and that's all made up of DNA?
Yeah, exactly. Your parents are going to determine your genetic features, and their parents determine theirs, and that heredity is the study of genomics. For instance, I have blue eyes and you have brown eyes. It's because our great-great-great-great-great-great-great-grandparents had genetic differences, and those differences arose from what are called mutations, or spontaneous changes in DNA. Those mutations, depending on whether they're good, bad, or neutral, will end up getting passed down from generation to generation, and we end up with different traits. In fact, we build all of the machines that sequence these DNA differences and mutations. Do you want to go check them out in the lab?
Sure. Do I get to our lab coat?
I think I can arrange for that.
Okay.
All right, let's go. So welcome to the lab. First thing we're going to need to do is get on some PPE, or personal protective equipment.
Got it.
Here is your lab coat as promised.
Thank you.
Inside the pocket, you're going to find some glasses and some gloves. All right, so welcome to the bench. This is where all of the science happens as far as getting your hands dirty goes.
Okay.
Basically, when you take DNA or any genetic material out of something that's alive, you're going to need to purify that DNA and clean it up before it can actually be read by the sequencer.
Okay.
So that's what we're going to be doing here, is what's called library preparation. Do you want to give that a shot?
I would love to.
Awesome, let's do it. All right, so after we've done our series of adding liquids to tubes and spinning down the tubes and adding more liquids and spinning, we finally get to the point when we can load our DNA onto the sequencer. And this tiny little piece of glass and plastic is called a flow cell. It has discrete channels, or paths through which that liquid can move, that have stuff on the surface that captures that DNA and allows it to be sequenced base by base.
Wow.
So every single base that's inside of that DNA sequence is going to be registered as a color or a piece of light by our cameras inside of those sequencers, and that will correspond to an A, T, C, or G, the letters that make up DNA.
Wow, that's so cool.
Yeah, it's crazy to think that this little piece of glass and plastic contains so much potential for uncoding genetic information.
It's awesome. Okay, so what's next?
What's next, for me, is the most exciting part. You ready to do it?
I'm ready.
All right,
let's go.
All right, here we are. We're at the final step.
Okay.
I'm gonna give you the flow cell, and it's gonna go right into this slot here. So what you wanna do is put your hands on either side of it and then place it into that little gap. Perfect.
There it is.
All right, you ready to press go and get this thing going?
I am.
All right, when you're ready, press that button.
It's so cool. I can't believe I just did real science. But what happens after this?
Well, it depends on the questions you might have as a researcher. One thing that we often look for are mutations or changes in the genome like we talked about. We have a reference genome, which is regarded as normal, and we can compare what we've sequenced versus that reference genome to see what might be different or has changed.
I've heard of mutations. Can they give me superpowers?
Well, if you think being able to drink milk and process milk proteins is a superpower, then yes. If you want X-ray vision, probably not.
Got it. But aren't some mutations bad?
Yeah, some mutations can lead to unchecked cell growth, where cells just continually grow when you don't want them to anymore. And that's basically cancer. There are some other genetic diseases like cystic fibrosis or sickle cell disease that have genetic underpinnings as well.
Got it. Can you only study genomics in a lab like this?
Well, you need a lot of the tools in this lab to do genomics as far as DNA sequencing goes, but as far as where the samples come from, your limit is really your imagination. For instance, I work as a volunteer in the Yucatan Peninsula of Mexico, diving into caves, collecting samples from there, and sequencing them so we can understand everything we can about the health of that aquifer.
That's so cool.
Yeah, so I mean, I know you're gonna be going to the Frozen Zoo at the San Diego Safari Park later today. That's a great place to get some genetic samples.
Got it.
The bottoms of the ocean, and even up in space. Really, the limit is your imagination.
Wow. Thank you so much for your time today, Chris.
Absolutely, it was a pleasure having you. Have a great day.
You too, bye.
Bye.
Nature isn't just full of smart designs. It's constantly tweaking them. Now that we know how genes and mutations work, I think it's time for us to see them in action. Some gene mutations, like the ones we just learned about, can lead to beneficial adaptations that help species survive and evolve. For example, check out the Antarctic icefish. It lives in the freezing waters around Antarctica. Water so cold, it could literally freeze most fish from the inside out. But this fish produces a special protein called antifreeze glycoprotein. Let's break that down. A protein is one of the key molecules our bodies use to build and maintain cells. And glyco means it has sugar attached. This protein helps stop ice crystals from forming inside the icefish's cells, which basically protects it from freezing. Scientists are studying these proteins to better understand how they might help treat human blood and bone disorders. Have you ever thought it'd be cool to live forever? Well, there's actually a jellyfish that kinda can. It's called the Turritopsis dohrnii, or easier to remember, the immortal jellyfish. It's super tiny, like the size of your fingernail. If it's injured, starving, or stressed out, it can actually reverse its life cycle and transform back to its earliest stage, like a biological reset button. It can still be eaten by predators, but otherwise, it could theoretically keep repeating the cycle indefinitely. That's seriously impressive. Scientists think studying it might actually help us learn more about aging in humans. And then there's the axolotl, a type of salamander that has an incredible ability to regenerate. It can regrow entire limbs, its spinal cord, parts of its heart, and even parts of its brain. No scar tissue, just clean, functional tissue, like new. Scientists are really interested in figuring out which genes allow this regeneration to happen, because it could lead to major breakthroughs in medicine. Gene mutations and adaptations happen in all environments, not just underwater. There are birds that can fly over the Himalayan mountains with little oxygen, to mammals with super healing powers or built-in disease resistance. Let's go behind the scenes and meet some of the experts helping protect animal genetics for the future. Hi, I'm Sloan.
Sloan, I'm Marco. It's so nice to meet you.
So nice to meet you.
You too, friend. I'm the wildlife ambassador for San Diego Zoo Wildlife Alliance, and we are in my favorite place in the entire world. This is a safari park. Do you know over 3,000 animals call this place home? It is wild out here. I say, you and I, it's time to have an adventure.
Let's do it. I can't wait.
Let's go.
Wow, this place is incredible.
It's wild, isn't it? Yeah, the safari park was considered to be the zoo of the future, where wildlife can really feel immersed in their native habitats. And honestly, all the work here at the safari park, whether it be a Sumatran tiger, maybe a platypus, all of that is tied into the work, say, with the Frozen Zoo as well. But right now, friend, I got something pretty special planned for you. I'm talking a safari truck in a savanna habitat. You might even see a giraffe, maybe even a rhino. Are you ready for this?
Yeah, let's do it.
All right, let's go, let's go. Sloan, I mean, look at this. This is amazing. East savanna habitat. So many different types of animals you would see out in Africa. I mean, even like the rhinos. Look at this, Sloan. Wow, these are southern white rhinos. Now, here at the safari park, we've had over 100 rhinos being born here.
Wow.
And that really speaks to the genetic diversity importance of what we do here. So we're part of what's called an SSP. It's basically a species survival plan. So we work with different zoos and aquariums here in the United States. So rhinos is a perfect example to help with that genetic diversity. And look, incredible, beautiful, vibrant animals. Wow. I'm a big fan of their eyes. Have you seen a giraffe eye before? Those big brown eyes?
Those big brown eyes.
How can you not fall in love with that? Wow. Giraffes here, they're often referred to as the watchtowers of the Serengeti. You kind of see why, right? They're one of the tallest mammal on the planet. And you know, what's interesting is we can learn a lot about giraffes and their physiology that can actually help out human beings as well. Now, it sounds really wild here, but their heart is about 2 feet wide. It can weigh 25 pounds, Sloan.
Wow.
That is the equivalent of about 50 human hearts, just an example. And talk about high blood pressure. They do have a higher blood pressure, and you think about it, think about why. I mean, the blood has to travel from the heart up about 6 feet of neck to reach the brain. Now, oftentimes people ask about hypertension or blood and water pooling around the ankles or the legs. Why doesn't it happen for a giraffe? Well, we've learned that actually the skin around the legs, it's almost like compression socks to help with swelling.
Right.
And nature has it built in. There's one more I gotta point out too. If you've never seen a giraffe drink, they spread their legs really wide, bring the neck all the way down. Have you ever gotten up really fast and almost felt lightheaded?
Right, yeah.
And you wonder, why doesn't it happen to the giraffe? Well, they have a special adaptation. So as you're lowering the head, the blood is actually gonna collect on the veins right before the brain. And then when they raise their head up, the blood flows back to the heart and it pumps that blood right away up to the brain. And there's still way more that we're learning, but that's one example of how wildlife can benefit humans as well, and just the beauty of Mother Nature.
So incredible. They're beautiful.
Oh, I agree. So friend, what'd you think so far?
This has been absolutely amazing.
Right? It's incredible, and you've seen so much, but now it's time to learn the science behind all the work they would do here and visit our frozen zoo. How's that sound?
I'm ready. Let's go.
All right, let's do it. All right, Sloan, so this is where the magic happens, and it is my pleasure to introduce you to Marlis Hauk. She is the curator of our amazing Frozen Zoo.
Hi, I'm Sloan. Nice to meet you, Marlis.
Hi, Sloan. Nice to meet you. Welcome to the Frozen Zoo.
Thank you. I'm so happy to be here. I've learned a little bit about the conservation work you do here, but how is this all possible because of the Frozen Zoo?
Well, the Frozen Zoo is amazing. I hope by the end of today you'll understand even more, but it was started in 1975, and it's the world's largest collection of skin cells and gametes. And these are living skin cells and gametes from all different species: birds, reptiles, mammals, amphibians, and a couple fish.
And how do you collect DNA from an animal?
We can collect it in a couple different ways, and it's usually after the animal dies. And we can take just a little skin biopsy, as long as the cells are still living, and then we process them in the lab, and they're in the lab for about a month while they grow and divide. And then we have to add a cryoprotectant. It makes it freeze kind of soft, like a popsicle, instead of like a hard ice cube. And so the cells are still alive in this frozen pellet.
Has there been a story of an animal in the frozen zoo that's really surprised you?
There is the po'uli. That is a Hawaiian honey creeper, a bird. They were rediscovered, I think, in the 90s, and they thought there were only 3 individuals, and that last individual died, and it was sent to us. And we were not freezing cells of birds at that time. We only knew how to freeze cells of mammals. Building on the techniques that we know, I just changed it a little and tried to apply it to the bird cells. And in the end, we were ultimately successful, and the cells are frozen in the frozen zoo.
Wow.
It's the first extinct species that ever had cells frozen from it.
That's so incredible. How is the frozen zoo helping to inform the future of human medicine?
A lot of different ways, but one good example is a researcher in Utah, and she's studying the effects of cancer in different species and how that could be applied to solving questions about human cancer. For example, whales and elephants don't get cancer, and so she's requested whale cells and elephant cells from the frozen zoo. Where else would you get those for your experiment? So we've talked a lot about the cells, we've talked a lot about the vials. Would you like to see what they actually look like in the tank?
Yeah, I would love to.
Okay. In this box, we know that there is a southern white rhino, frozen. And so, what do you think about holding this?
I would love to.
Okay, go ahead and put that glove on. Okay, and we'll stay over the nitrogen in case you dropped it. I keep dipping it to make sure it stays frozen. But I'm gonna put that in your hand. So right now, you are holding a frozen pellet of 1 to 3 million living southern white rhino cells.
That's so incredible. Wow.
And then the best part is when you put the lid on. Watch this. Yes, ready?
Nature isn't just full of smart solutions. It's constantly inspiring new ones. We've seen how studying DNA can help protect animals, but nature also gives us ideas for solving all kinds of human problems. For example, have you ever been bitten by a mosquito? You probably didn't even feel it, right? Scientists studied how a mosquito's proboscis works and is used to design a painless needle for medical use. Researchers looked at the tokay gecko's super sticky foot hairs and used that idea to create a special adhesive that seals wounds. No stitches needed. But nature's inspiration goes beyond medicine. There's even a paint inspired by the lotus leaf. When water hits the leaf, it rolls right off and takes the dirt with it. So, scientists made a paint that actually cleans itself when it rains. And did you know that sharks don't get barnacles like other sea creatures do? Ever wonder why? Turns out their skin has a unique texture that repels tiny organisms. Scientists mimicked that texture and used it on ship hulls, making ships faster, cleaner, and more fuel efficient. Nature isn't just cool to look at and learn about. It's basically the world's smartest design lab. It's pretty amazing what we've learned by just studying life right here on Earth. But what if we took that curiosity even further? Believe it or not, scientists are doing real genomics research about space. Let's head to NASA's Johnson Space Center in Houston, Texas, where my co-host Christian is standing by to help explain how studying genes and microgravity is opening up a whole new world of discovery.
Thanks, Sloan. Today we're going to meet scientists who are studying the world of genomics from right here and from space. Come on.
Hey, Christian, come on in.
Hey.
Welcome to the microbiology lab at the NASA Johnson Space Center.
Wow, this is so cool. So you helped pioneer the ability to study genomics in space.
I did, yeah. My team and I put a DNA sequencer on board the space station in 2016. Come on, let's grab some lab coats and I'll tell you more about it.
Absolutely.
So yeah, we had more than 14 astronauts do more than 60 sequencing experiments to date. Once we started sequencing on board, we haven't stopped. And we're using it for things like the ability to identify microbes in the space station in real time, as well as many more things I'm excited to tell you about.
Wow, I'm ready.
All right, let's go. I want to introduce you to one of our lead researchers that was critical in enabling method development for sequencing in space. This is Sarah Rommel.
Hi, Sarah. It's nice to meet you.
Hi, nice to meet you too. I was just looking at some plates from the International Space Station. Before we had sequencing capabilities in flight, we used these culture-based techniques. Would you like to see some space bacteria?
Absolutely.
All right, grab some gloves. So here's some bacteria that we got from the surface on the space station. It's not dangerous. It's probably just some bacteria that came off the skin of an astronaut.
Oh.
Yes, if you want to look at the microscope, we have some gram stains in here. And this is a way that we can identify some information about the bacteria to see if they're gram negative, gram positive, or bacillus.
Wow. Now this is cool.
Yeah, you see lots of tiny little like purple rods in there.
Purple rods, little dots.
So we can use the microscope to get information about it, but sometimes it's not enough for us to identify what it is. And so that's why we use DNA sequencing to get additional information to identify them.
Now, I have to ask, have y'all been to the International Space Station?
Sadly, no, but our voices have been. Every time an astronaut is doing our sequencing on board, either Sarah or I are talking to them and talking them through it.
Oh, that's really cool.
It is really cool.
So, how do you sequence DNA in space? And what's so different about it than doing it on Earth?
So you can see in the laboratory on Earth, we have access to all of this hardware, all these different reagents and trained personnel. But in spaceflight, we have to be much more limited in the hardware that we can fly, the reagents that we fly, and the kind of experience level that they have with pipetting and working the lab. But that's why it was so important for us to create a streamlined protocol. So we make these one-time use kits to be able to quickly complete the process. And we also are able to communicate with the crew and talk to them while they're doing it.
So, what is a reagent?
A reagent is a chemical that we use to complete our science. So this is a reagent. This is magnetic beads that we use to clean up our DNA. So we developed this instead of using on the ground like you'd use a centrifuge that's too large for us to use in spaceflight. We can add the DNA to this and it will bind to the DNA. And then when you put it on a magnetic bead stand, the DNA and the pellet will form against the beads. So you're only left with clean, pure sample to go forward for sequencing.
Oh, wow. How can studying genomics in space help with human health?
Every time we put a microbe in a new environment, we learn something about them because they adapt. And we do the same thing. We adapt and we survive and thrive. And so there's parts of our genome that we don't really understand. Being able to study genomics in space, we would see parts of these genomes start to be expressed. And that would tell us that they're providing some kind of advantage. Learning something about that would also teach us new ways to treat or cure a disease. So with humans, who knows what it would unlock, but there's a lot of possibility.
Now that you've had experiments in space, what do you really hope to work on next?
So what we're really focused on after ISS is going to the moon and onto Mars. So one of the things that we're really concerned about with Mars is what of our microbes are we putting on Mars? And we're not going to be able to help it, right? If we someday are to find signatures of life on Mars, we want to know that it came from Mars and not from us.
Wait, you might be the first person to see DNA from another planet. How does that feel?
It's incredible that we have this technology and we're even talking about this. So this technology and DNA sequencing will be one part of the bigger puzzle to characterize what is life, right? But if we found something like DNA, it would sure be a signature of life. So it's really exciting to think that we're, getting closer and closer to that, and that we have the technology to be able to do that in place without even having to send samples back to Earth. So a lot of exciting things to come.
So cool. Now, how do y'all communicate with the astronauts in space?
We could tell you, but I say we just show you. Do you want to see?
Totally.
Awesome. Well, come with us to our mission operations room.
Okay.
All right, here we are. You ready to see DNA sequencing in space?
Of course.
Awesome. Hey, everybody. And here's a procedure for you to follow along.
Thank you. All right.
All right, we are getting ready to go live to the ISS with astronaut Don Pettit to help him prep some samples for DNA sequencing on board station. So Don, we're gonna go ahead and have you pipette all of the extracted DNA into the magnetic bead tubes, and so that we can purify that DNA for downstream sequencing.
Will do.
And those are the same magnetic beads we saw last time, right?
They are the exact same ones you just saw.
I'm going to give them a little flick to get all the stuff down to the bottom of the tube.
So why does he have to shake it so hard?
So in the lab, we want to make sure all the liquid is at the bottom of the tube. In space, you got to use a little more force to help force the liquid to the bottom of the tubes.
Just to make sure, I've got the pipette set to 20 microliters. I've got a bunch of tips ready to go in case I need.
And copy that. That was a great readback, Don, and you are go to carry out that next step of the reaction. So Don's gonna keep working to get these samples purified and ready to go on the sequencer. And even though we've been doing this for a few years, you can tell we're all still really excited. You know, when we were showing you those cultures, it would take us months to get those IDs back. With this, we know what those microbes are within hours. So a huge increase in the time from sample to answer. So this is definitely the future.
Wow, I'm really impressed with the work that y'all do here. We've learned so much about being in the lab, working with astronauts, and also discovering the next frontier of genomics.
It was great to have you. Thanks for being here with us, Christian.
Well, thanks for having us.
Yeah, absolutely. All right, Don, let's finish up getting our samples ready to be loaded into the sequencer.
Thanks for joining us on our Nature's Blueprint virtual field trip today. I want to thank all the amazing experts that we met along the way, and of course, my amazing co-host, Sloan. Now, we challenge you to dig into a genetic trait that you're curious about and see how nature's teaching you a little bit more than you realize. On behalf of Discovery Education and Illumina, I'm Christian. I'll see you next time.